In chapter 6, we're going to talk about chromosome variation. This is generally going to be structural variation, so changes in the overall structure of the chromosome, or it's going to be changes in the number of chromosomes present in the organism. Now, generally, any major changes in structure, number, or variation are going to have a negative phenotype on an individual. But quite a bit of evolution has happened because of chromosome variation of one sort or another. So there's an example up here on the top right. Ancient gene linkages support ctenophores as sister to other animals. This is an article you could look up if you would like. You can just use this information here to Google this. This has to do with animal phylogeny and the closest most ancestral group to all of our animals. And we determined that the ctenophores, which are comb jellies, similar to jellyfish that you would be familiar with, but have some very specific characteristics, are actually a separate group from the jellies and are a more ancestral group based on some specific chromosome variation changes that are unique to that group. So as we go through chapter 6, we're going to talk about the evolutionary significance, and we're going to talk about some examples of problems or disorders, diseases that might come up from changes in our chromosome structure. Our textbook opens with wheat as an example of chromosome variation, and it is an example of something called polyploidy. Polyploidy is when we have multiple copies, more than 2 copies, of an entire set of chromosomes. So we've talked about haploid, where organisms have one set of chromosomes. I shouldn't say organisms, I should say cells, usually our gametes. And then we have talked about diploidy, where we have 2 copies of each set of chromosomes. Now, something that's unique to plants compared to animals is that they can be polyploids, meaning they have more sets than just 2. They could be triploid, they could be quadruploid, pentaploid, hexaploid, anything you might want. Strawberries, for example, are octaploid, meaning they have 8 copies of all of their chromosomes. Now, this can come up in a variety of different ways. Wheat being hexaploid is our example here. And if we follow this example here, we see that we start out with a diploid species and a diploid species. And throughout the hybridization of these 2 different groups, there was a non-disjunction event, meaning that the chromosomes didn't properly separate. Now, this immediately causes speciation because chromosomes are not compatible with either one of the parents. Then they were crossed with a wild grass and acquired a 3rd set of chromosomes, resulting in 6 copies of each chromosome. So we'll talk about this a little bit later, but this is just sort of our introduction to what we might mean by chromosome variation. We're going to go through a couple of different types of chromosome variation, but we need to start out by talking about why chromosome variation is important. And that's because every species has a characteristic set of chromosomes. They have a distinct number of chromosomes, and each one of those chromosomes has a particular size and shape based on the genes and the number of genes that are residing on that chromosome. So for example, humans are diploid with a total of 46 chromosomes. Each one of our chromosomes has a characteristic size, shape, banding pattern on a karyotype, order of genes on a chromosome. And that is going to be the same for all of our humans with a normal complement. And the reason that we use the term species here is that we generally define a species by what we call the biological species concept. And the biological species concept says that 2 organisms or 2 populations, 2 groups, are the same species if they can interbreed and produce viable, which means healthy and living, viable and fertile offspring. Now, fertile is important alongside viable because if we're talking about genetics and if we're talking about evolution, we need our genes to be passed on. So if they die before they pass on their genes, they're not contributing to the next generation. And if they are infertile, they're also not contributing to the next generation. And so if we cannot pass on the genetic information for this group, they're not considered species. The reason that we're talking about this here is that we're going to be looking at the distinct chromosomes in some of these organisms, or at least organisms in general. And if their chromosomes are incompatible, the cell cannot undergo meiosis properly. So we get improper chromosome alignment in meiosis. So what this means is that our individuals are not going to have proper chromosomes for them to line up and separate. And so assuming that they survive to reproductive age, they're not going to have gametes that are going to be compatible with other gametes because the chromosome number or complement is going to be incorrect. So we're going to talk about some changes that can happen. Keep in mind that we could also have mutations, but mutations are not changes in chromosome structure. They're changes in chromosome sequence, and we will get to those later. We're going to start with talking about changes in structure, such as gain and loss of pieces or changes in gene order. Then we're going to talk about variation in number. So this can be number of a single chromosome, like in the case of Down syndrome, which is what we would call a trisomy, meaning that there are 3 copies of one chromosome, specifically chromosome 21. So another name for Down syndrome is trisomy 21. We could also talk about those extra sets, extra entire sets, and that was our polyploidy that we talked about in our example. So we're going to talk about both aneuploidy, which is that single chromosome change, and then also polyploidy, which is the entire set change. So as I mentioned in the intro slide, this is also really important for evolution. So we're going to talk about some examples of when we've seen evolution or when we see disease and things like that for each of these variations. So before we get going, we're going to review 2 things really quickly. One is chromosome morphology. And this is important because we're going to be talking about changes, and many of these changes are going to be relative to the chromosome. So just a reminder, chromosomes with centromeres in the middle are called metacentric. Submetacentric basically means away from the center a little bit towards one of the ends. Acrocentric means almost the end, but not quite. And then telocentric means that the centromere is on the end. Remember that the short arm is called the P arm or the petite arm. And on this metacentric, we wouldn't have a petite arm, but on all of the other ones, at least submetacentric and acrocentric, we have that petite arm. And then telomeres don't have a short arm. They only have the Q arm, which is the other arm on the chromosome. Q stands for long. I don't know why. Just somebody decided to name it the petite arm and basically the not petite arm, and Q becomes next. So we get the P and the Q arms. We look at chromosome structure using a karyotype. Karyotypes are made from cells that have just entered metaphase. So they chemically treat them so that mitosis is paused, and then separate or break open the cell, release all of the chromosomes, and then take a picture of it. And then they use software to line up, put them in nice, neat rows like we have here, so that you can compare the physical structure of our chromosomes. So chromosomes are aligned according to size. So our smaller numbers are going to be our larger chromosomes. So they're aligned largest to smallest. So the largest chromosome in any organism is going to be chromosome 1, second largest chromosome 2, and then of course we'll go all the way out to the smallest one is whatever the last number is. So in humans that happens to be 22. You can see that these last two chromosomes are really pretty tiny. And then finally, we have our sex chromosomes. In this case, this is a biological male because we've got an X and a Y. And those two are considered homologous because they have special regions that allow them to pair during meiosis, even though they don't really follow the rule of having the same size, shape, and gene order. Our chromosomes are stained with several different types of staining depending on the purpose of the stain. Their stain works because different regions are condensed at different levels. So the most common one is called the Giemsa stain. And the Giemsa stain is going to stain AT-rich regions. And you'll notice that this gives a particular pattern. And if you look at our homologous chromosomes, they have the same pattern of dark staining. And this is kind of what we mean by same gene order. These stains do not reflect genes, but they do reflect specific structures within our chromosomes. So their specific sequences are going to allow specific packing, and the more tightly packed, the darker the band we get. So I usually think of these as this is kind of the bands are the equivalent of the same gene order, same banding pattern. So instead of saying same size, same shape, same gene order, we could also say same size, same shape, same banding pattern. And that banding pattern is also going to help someone to recognize if there is a change that has resulted from a chromosome variation. So if a piece of a chromosome is broken off or something like that. So we are going to look at these different chromosomal rearrangements. I like this diagram because it directly compares what's happening with our chromosomes. For rearrangements, we are going to talk about duplications of specific genes. We are going to talk about deletions. I shouldn't say genes, I should say regions, but there will be genes in those regions. Duplications, deletions, inversions, which is where a region of a chromosome is flipped over where it's supposed to be. And then translocation means that we get a piece that is moved to somewhere else. So either on another chromosome or the other end of the same chromosome, something like that. Then, of course, we've got aneuploidy. This alters the number of one specific set of chromosomes. So this is only going to affect one type of chromosome at a time, at least as far as it is survivable. And then polyploidy is our extra set of chromosomes for every chromosome that we have.